Okay, welcome to the Carey Institute Thursday seminar series. My name is Emma Rosie, and I'm going to introduce our speaker. And first, I want to welcome everyone who is here online. And of course, all of you are here in person. It's nice to see you all. Our speaker today is Dr. Jess Brandt. She's from University of Connecticut, where she started in January of 2020, a challenging time to start your faculty position. Before that, she did her undergrad and master's in public health at Johns Hopkins. And then after that, she had an interest in kind of shifting gears a bit and working more on contaminants and how they affect ecosystems and vice versa. And so she did her PhD at Duke University, co-advised by Emily Bernhardt, who many of you know, and Rich DiGiulio. And then after that, she went and did a postdoc at USGS in Fort Collins, and a bunch of her work today, I think, is going to focus on some of that. And Jess's research, for those of you who know me, it's really close to my interest, and I'm really excited to hear what she has to say. She's really interested in the intersection of contaminants and ecosystem processes and how ecosystems affect contaminant transfer as well. So thank you so much for joining us today, Jess.
Okay, I think my microphone's on. Can you guys hear me okay? Oh, there we go. Okay, all right, sorry about that. Thank you so much for the kind introduction, Emma, and I'm really honored to be here and have the opportunity to visit the Carey. What a nice facility and group of people. So before I jump in, I want to acknowledge that I'm at the University of Connecticut, and UConn is located on the territory of the Mohegan, Mashantucket, Pequot, Eastern Pequot, Schaghticoke, Golden Hill, Pawcasset, Nipmuc, and Lenape peoples who have stored the land throughout the generations. The title of my talk is The Role of Aquatic Food Webs in Contaminant Cycling, Transport, and Toxicity. Hopefully the slides come out okay. We had a little bit of weirdness, but I'll start with just, when I was sitting down to think about how I wanted to frame this talk, it gave me an opportunity to think, reflect a little bit on how I arrived at this position in my career, how I see myself as straddling the ecology, ecotoxicology, ecosystem science boundary, and kind of how I arrived at this place and how I see my research program. So I want to share a little bit about that, and that's how I'll start the talk and frame the talk. And following that, I'll jump into two case studies, one about selenium trophodynamics in the lower Gunnison River Basin, and the second about salmon biotransport that sort of show a single ecosystem and then trans-ecosystem transfer. Okay, so I see contaminants as my study organism, and naturally you would want to start a talk introducing your study organism, but that was a lot harder to do than it seemed. I flipped through 4 textbooks on ecotoxicology that I had at my desk, and none of them had a definition of contaminants. Like, wow, okay, maybe we should have thought about that. And so I Googled it, and Google tells me it's something that contaminates, and that's really not helpful. If you look at Jason Rohr and Kirby and C's 2006 paper in Trends in Ecology and Evolution, they provide a definition that's pretty broad, and they define it as abiotic stressors. Maybe that's a little broad for my purposes. So the definition I'll work with today is chemicals that occur where they wouldn't naturally, or at higher concentrations than they would occur naturally. So as Emma mentioned, I started in the public health space and was thinking about toxicology as it was derived from the field of pharmacology, very much human health perspective on that. What do contaminants do to people? And so toxicity and toxicology are defined similarly, the adverse effects of exposure to chemicals for biota, and toxicology being the study of those adverse effects. And then transitioned with my PhD to be thinking a little bit more about sort of what does the environment do to contaminants. I was really interested in sort of contaminant mediation and how that affects exposure and the effects of exposure. And I was working at first in an ecotoxicology lab. Ecotoxicology is defined as the science of contaminants in the biosphere and their effects on constituents of the biosphere, including humans. So what all these definitions have in common, in my view, is that they define the agent as the chemical contaminant and the recipient being the biological system. But then I started working at the crossroads of 2 labs, and I was working with Emily Bernhardt's group, and it's an ecology group and a biogeochemistry group, and really transformed my thinking about contaminants as materials in the environment that don't have an agenda, right? It's just a chemical, it doesn't have a purpose, In the sort of, if we wanted to humanize a chemical, it's not set out to do anything harmful. It's really the biological system that determines what a contaminant does. And so I started thinking about this a lot more so as a reciprocal interaction, not a one-way interaction, but a reciprocal interaction between a chemical constituent and the biology that it interacts with. This sort of framing is central to my talk today, and it really transformed how I started seeing myself and asking questions about the environment and ecosystems. And so once I started thinking about it this way, examples just started pouring out of the literature. And so some that are just readily available, we think about processes like uptake and biotransformation, distribution, retention, excretion. All of these, in my view, are biologically driven processes. Here's just some images, and these really span multiple levels of biological organization. So we're looking at sort of uptake into the food web, then we've got cellular transformations, chemical transformation that happens at the cellular level, and partitioning that happens at the cellular level. This also happens at the food web level. The food web controls kind of what forms of chemicals get into the system, how they change as they're in the biological system, which ones get transformed or transported to the next trophic level. And this can also apply across ecosystems. So we think about paired ecosystems and paired food webs, like the stream to riparian food web, for example. Okay, so if you're with me now on the same page, this is how I think about contaminants, and I want to talk about two stories that I think highlight this thinking pretty well and demonstrate the power of this thinking or the value of this perspective. And my goal will also be to highlight some of the work that is building on this work and areas that how we're kind of furthering these themes as I go through the two case studies here. So when I started my postdoc at the USGS in Fort Collins, one of the projects that I was brought on to work on was this really beautiful data set covering selenium trophodynamics in the lower Gunnison River Basin. So this reflected 2 years of really intensive food web sampling and abiotic compartment sampling that was done before I got there. So the data was kind of ready and in hand, and a bit of a mess, but that's okay. Pulled it all together, and this work is just out last year. And I want to acknowledge that James Roberts, Craig Stricker, and Travis Schmidt, all at USGS, were really active collaborators in this process and did a lot of the sample collection and data processing. And then Holly Rogers and Patricia Neese were students at the USGS at the time and have since moved to other institutions. I'll start with just an introduction to selenium. So it's in group 16 of the periodic table, and we don't need to go through these one by one, sorry about that. Discovered a long time ago, it's a chalcogen, and it's decidedly a non-metal. I know it often gets described as a metal, but it's not a heavy metal, it's a trace element and really a metalloid. And the key distinction there is that it's an oxide anion in solution. So its mobility increases with pH. In the environment, it commonly occurs in both inorganic and organic forms. And a key thing to know about selenium is that it is required, so it's an essential element, but it's also a contaminant. And there's this really narrow threshold of concentration that takes it from being beneficial to harmful. And the reason that it's essential is because selenium is a key component of selenocysteine, which is the 16th amino acid. And that amino acid is then incorporated into a huge variety of different proteins, maybe most famously are the antioxidant enzymes, some of which are shown here, the glutathione peroxidases, thioredoxin reductases, and the iodothyronine deiodinases. So selenium is a hell of an element to start thinking about biogeochemistry from because it's super complicated. So it was a really fun, I think, introduction, but it was hard coming from a very different way of thinking. It occurs in the environment, as I said before, in both inorganic forms and organic forms, but it's loaded into the environment. And if you went and measured a water sample, you're going to find it either primarily as selenate or as selenite. And what determines how it occurs really depends on sort of its source. Is it geologically sourced, or is it sourced from, I guess, which also would be geological, but different fossil fuel substrates? The things that you want to keep in mind when you're thinking about selenium in the environment is that the loading rate can really vary seasonally and anthropogenically. So there are different patterns depending on what system you're focused on. We know now, after decades of research, that food web assimilation is the most important component that selenium cycling is really biologically mediated in the assimilation into the food web is the critical step in that, but it's really poorly understood. And I'll highlight some of that lack of understanding or gaps in understanding a little bit. We know that water concentrations are very poor predictors of food web concentrations. We've assumed for a long time that that hydrologically speaking, low water, long water residence times allow for greater uptake. It's been thought that selenium is in the system for a prolonged period of time, and that gives it more opportunity to get into the food web. And so we've thought that selenium was more of a problem in reservoirs or lakes than it was in rivers or streams, for example. And in terms of partitioning, we know that selenium sorbed or absorbed to the particulate fraction is the primary route of food web entry. In 2016, EPA revised its criteria to really emphasize tissue concentrations or food web concentrations rather than abiotic concentrations like water concentrations, which is its norm for so many other types of constituents in waters because of how important the food web is in determining what effect selenium might have to organisms. So this project is situated in the lower Gunnison River Basin. The shape of the watershed is shown here, and the shape of the basin. And this is where it's situated in the broader upper Colorado River Basin in the top left. And underlying the Gunnison River Basin is the Mancos Shale. So this is the geological source of selenium in this system, naturally enriched in selenium. And it's really the chemical weathering that happens to expose shale, but also irrigation waters exposed to this landscape to water crops, then carries and mobilizes a lot of that selenium into the river. This river is impaired, and the lower segment, which is highlighted in orange in this lower segment of the Gunnison River Basin, is federal critical habitat for several species of fish, including the Colorado pike minnow, which is maybe the most important culturally and ecologically in this system. It's a very long-lived migratory species, and its numbers have been greatly reduced. And so the motivation for this project was, what do we know about selenium in this system? How does selenium change in this system in both space and time as we move through this? And the reason we wanted to focus on the temporal question is, are the periods of exposure highest during sensitive periods for this fish species, so that we can sort of understand better when to be monitoring selenium and what are our benchmarks for monitoring? What are we trying to get below, and when do we need to be sampling to make sure we're doing that? So this is a really dynamic river system. You can see here just the hydrograph from 2015. In blue, you're looking at discharge in CFS of over a calendar year. So starting in January and moving to the end of the calendar year. And overlaid on that is the gray line, which shows the predicted selenium concentration. There's one gauge at Whitewater that measures selenium concentration pretty routinely, but it's primarily modeled off of the particulate concentration and the conductivity. And the bottom graph shows the probability of exceedance. And what I want you to notice in this figure here is that in these summer months, when flow rate is highest, it's assumed that selenium concentrations have the lowest probability of exceeding these criteria levels. Other times of the year, selenium is going to be higher, and that's, again, following up on this assumption that it's hydrologically driven, and a high water flow means dilution, and so there's going to be less selenium in the system, less of a concern. So the paper did a couple of things. We built a food web model for the system, and we looked at various types of particulates and how they predicted selenium relative to measured concentrations, and arrived at the conclusion that green algae gives us predictions that are most similar to measured concentrations. So we've identified our kind of key food web component, sort of the point of entry for the food web. And then we built a food web model using stable isotopes to understand sort of the relationships. And using this information, we then modeled the rest. Sorry that the figures are blurry. Modeled the trophic relationships that we're interested in. And this is what our food web model looks like. I'm not sure if folks here are familiar with the Presser and the WOMA 2010 ecosystem scale model, where they pulled data from all over the world. This is a data set that we sort of adapted that model to fit this ecosystem. And the model is actually quite simple. You look at your various components. The x-axis is showing your sort of food web steps as defined by our model, and selenium concentration shown on a log scale on the left. And you have both water concentrations in milligrams per liter and biotic or solid phase samples represented in micrograms per gram concentrations. And the different metrics here, the KD, which is our partitioning coefficient, and then the trophic transfer factors are really just ratios of concentrations between the two things. And this is standard ecosystem scale modeling approach for environmental selenium. And we still see, even with the robust modeling effort, we still have really robust coefficients of variation. Water's very similar in terms of variation to the rest of the food web, and we're still seeing, even though we know that green algae is the best fit here, a lot of uncertainty. Okay, so some other things to know about this system. High dissolved selenium concentration, super high. So they're routinely exceeding EPA's aquatic criteria of 3.1 micrograms per liter for lotic systems, which is a concern, and that would be enough to designate this system as impaired. We know that there's really efficient uptake. So when we look at the KD value here, the partitioning uptake, it's higher than we typically observe in lotic systems for selenium, and it's similar to what we would observe in sites that are impaired by mining or in estuaries and ocean sites. So this is kind of a unique river system. And the high tissue, and then we also observe really high tissue concentrations. So Hamilton in 2003 introduced the idea that a 4 microgram per gram dry weight whole body concentration is our benchmark for toxicity for fish species, and 91% of the speckled dace, which is our low trophic level fish species in the system, exceeded that criteria, and 96, almost all of the round-tailed chub. We measure these two species of fish because the other species are so sensitive that there's just really no political will to allow for sampling of those fishes, and these are fairly good proxies in terms of diet and food web position. The two things I want to point out here that really clued us into just how influential the food web is. Here are the selenium concentration in the different fractions. So here we've got dissolved in blue, and we have two different particulate fractions, green algae in the hollow green circles, and the total suspended solids in orange. We're looking at concentration on the top row and partitioning the ratio of water to particulate concentration on the bottom row over the time series of October 2015 to October 2016. So 4 sampling time points in this 1-year period. And it's really in April 2016 that we observe this bizarre behavior. This is the high flow period in this system. Snow melt driven releases of water to the system occur. And this is when we would expect, dissolved concentrations are relatively low compared to other parts of the year, but the green algae concentrations are super high. So this is puzzling. We don't know exactly what's going on here, but there's a lot of water coming in. The concentration of selenium in that water is low, and yet the green algae concentrations are higher than they are at any other point in the year. And that's reflected also in the partitioning coefficient. So we say now that green algae has the ability to sort of shunt, if we take the pull shunt concept and apply it to the system, shunt selenium out of the water column and into the food web. And that's happening in April. And then the other interesting pattern, temporal pattern that we observed here is how the upper trophic levels then respond to that high algae concentration. So in April, high algae concentrations are matched by high caddisfly concentrations, as you would expect. The caddisflies are directly eating the green algae, and their concentrations match, and the trophic step increase is what we would expect. It's a 1.3. But then in August, when we went back and sampled, the green algae concentrations fell down to sort of our baseline base flow type scenario, but there was really no change in the caddisfly concentration. So the caddisflies are then holding on to these high selenium concentrations. That's puzzling. We think, it's likely a diapause type process. We don't know for sure, but this is interesting. If you think about how managers manage these systems, they go in and they measure the dissolved concentration and they estimate risk based on that. That's not explaining any of these patterns. And so this is really a compelling case that we shouldn't be measuring that at all. I would say if managers are going to go into these systems, they should be managing for concentrations in the invertebrates. If we take this together though, and then extend it to the rest of the model, we can predict that the concentrations of selenium are going to be highest in the rest of the food web and in the species of concern. These are estimated or predicted concentrations from the model. They're going to be highest in April 2016, April and August over the calendar year. And why that's concerning is that coincides with these periods of highest these really sensitive reproductive windows. Selenium's a threat to animals that lay eggs because it is enriched in yolk. And so animals that rely on yolk-based sustenance in the early parts of their lives are getting a huge hit of selenium. And so that's true that these periods of time are when egg development prior to spawning, and then early hatch and release into the ecosystem. And so, yeah, this just brings back that point that we're measuring the wrong thing and we're thinking about the wrong time periods. And if we're managing the system for the protection of fish, we need to be thinking about what fish are eating and when they're being exposed to high selenium. We're expanding. This data set was not just a selenium data set, though that was the motivation for collecting it. We measured a whole bunch of other elements in the food web at the same time. One of them was mercury. Maybe this audience is familiar with the hypothesis that selenium has this potential to mediate mercury sort of methylation at the base of the food web, uptake at the base of the food web, but also toxicity. There's a lot of interest in that. And so we're building off of this paper and a perspectives paper that we wrote outlining all of the kind of gaps in understanding about selenium mercury interactions to ask, are selenium and mercury coupled in this system? And PhD student Raul Flamenco in my group is leading that work. There's also a whole bunch of other research that we want to follow up on. So selenium is a contaminant, an element that I've been working on for about 10 years, and I'm really interested in continuing to do so. And so we're building a research program that will ask questions ranging from sort of what are the abiotic versus biotic controls on uptake. This is that sort of the key part of uncertainty in food web cycling, but it's also the most essential and the highest concentration enrichment that we observe in any of the steps. So we really need to understand what's going on there. What are the abiotic versus biotic controls? Which ones are more important? We're also interested in thinking more about selenium redox physiology. It's an antioxidant, but at high concentrations, the leading hypothesis is that its mechanism of toxicity is an oxidative stress mechanism. So that means that selenium sits sort of on this redox teeter-totter. And what accounts for that switch? When is it toxic? When is it a nutrient? I mentioned just a minute ago that we're asking a lot of questions about how selenium and mercury interact. And so we've got Raul's project that I just mentioned. We're thinking about what are the implications for adopting selenium mercury molar ratios as a metric for public health instead of the mercury-based advisory metrics that we're using currently. And we're also thinking about how primary producer communities are driving this, because different primary producers have varying sizes of their selenoproteome or requirements for selenium. And then finally, the Gunnison is a really good case study here. There's another one in the Cucanusa system in Montana, spanning the Montana-British Columbia border, about how selenium might be constraining these broader efforts surrounding fish conservation. Okay, so now I'll transition into this next case study. And we've just talked about sort of one ecosystem and the food web over time. Now I'm going to be talking about transboundary movement of subsidies between the ocean and freshwater systems and how Pacific salmon mediate that process during their migration. My collaborators on this project are Jeff Wessner, Tim Jardine is at the University of Saskatchewan. Gabby Russo was a student that got this work started, is now at U Missouri pursuing her PhD, Chris Voss at Regis, and then Colin Eaglesmith, Craig Stricker, and he got cut off on here, but maybe the most important collaborator on all this is David Walters, who called me when I was thinking about joining them for a postdoc and was like, Jess, how much mercury does salmon bring back to the continent every year? We want to know, come figure it out for us. I was like, okay, that sounds awesome. And that was the genesis of all this. Gabby had started working on this data. So maybe this is quite familiar, but for those it's not, Pacific salmon have this really compelling life history. They begin their lives in freshwater systems and spend some variable amount of time in growing and maturing in those systems before they migrate out to the ocean. When they're out in the ocean environment, they accumulate the majority of their biomass, more than 95% of all of their biomass, also more than 95% of all the subsidies that they're going to bring back to freshwater systems. When they're ready to return to spawn, they do so. They travel hundreds to thousands of kilometers back to their freshwater systems, the same systems they were born into, and spawn, and then they spawn once, and that's the end of their life cycle. They die after that. they undergo a pretty incredible physical change with this. This is an ocean, pretty sure this is sockeye salmon. This is an ocean phase sockeye, and this is a freshwater phase sockeye. So they undergo this sort of, I'm not sure if it would be classified as a true metamorphosis, but a physical change. And when they hit that freshwater boundary, they don't eat. It's assumed that they meet the freshwater boundary, and at that point, they're just sort of on a mission to get back to their spawning grounds, and nothing gets in their way and distracts them. And that's important for this study because we're trying to quantify the flux from ocean to freshwater, and that would get muddied if they're eating a bunch of things in the freshwater system too. But they really catabolize their energy stores from the ocean during this process. And here's just an image showing the spawning grounds, and you can see the beds, the reds that they build for spawning purposes, and the blue circles are highlighting carcasses, salmon carcasses. So just to kind of reiterate that they do die just after spawning. Okay, so the first result of this project was quantifying the number of fish. 123 million Pacific salmon per year escape to freshwater spawning grounds. What do we mean by escape? We call this process escapement, the number of fish that actually make their way into freshwater systems. And I like to think of it as they get past the commercial fishery. So they're literally escaping the commercial fishermen. And these are the 5 species that we're thinking about. These are the semilpara species, the ones that spawn just once. And they're Chinook, coho, sockeye, pink, and chum salmon. And this is when they return to North America, this is their geographic range. So as far as the sort of northern reach of the Bering Sea, and then all the way down to, actually our data set extends down to the runs that enter into California waters. When they get back to systems, ecologists have been studying this for years. The nutrients that they store or release from their bodies are used for a huge number of processes. These nutrient fluxes influence both bottom-up and top-down processes, and this paper by Gend outlines in a really nice way, I think, all of the various routes that those nutrients are used. Are they directly consumed, or do they decompose in the stream bed? But they also there's been more recent emphasis in that they don't just transport nutrients, they also transport contaminants. And really in this framework that I employ, the nutrient versus contaminant dichotomy is a little bit arbitrary in my view. The reason we're thinking about them together in this case study is that they explicitly share pathways of transport. So all of the materials are derived from the ocean. They partition at the base of the food web and build up as they move through the food web. They then transfer to other organisms. Here our receptor species is migratory fish. When they are transported, salmon go from being sort of diluted in their spatial distribution in an ocean and then migrate, and they concentrate in high numbers in relatively small areas. So there's sort of this focusing of subsidies when they get there. And then both nutrients and contaminants are released to these freshwater systems in the same way. They're deposited, or they're transferred through consumption, or they're recycled through decomposition and excretia, for example. And the final piece of background here is that there's been a lot of concern about what's happening to the Pacific salmon community. You can't go a week without reading some article about numbers are falling or the size of these fish are decreasing. There's this pretty interesting take on, what are the consequences of smaller fish size, and now wholesalers won't buy salmon from commercial fisheries because they're too small and they don't meet market expectations. And a paper by Oke et al. Just recently in Nature Communications quantified what does the decline in fish body size mean for subsidies? What are the ecosystem consequences, recipient ecosystem consequences of smaller fish coming in? So right around this time that we were getting started on this work, Greg Rigoroni, who's a salmon fisheries scientist at the Natural Resources Consultants, and his co-author James Irvine published this paper where they quantified both numbers and biomass of ocean-derived salmon. And we reached out to them and said, hey, you have this ocean abundance. How hard would it be to pull out escapement numbers from that? And he said, oh, it's actually pretty hard, but let me try. And so he did. And while he did that, we asked him to focus on just North America. So we excluded the Asian stocks from our analysis. And we also added in the other 2 species. So they were thinking just about Chinook and chum. Sorry, they were not thinking about Chinook and coho. They were thinking about the other 3 species only. So they rounded out the number of species they were considering and gave us escapement numbers. And that ended up constraining our data set a little bit. So theirs was a 90-year analysis. We are working with a 40-year analysis from 1976 to 2015. There were three motivating questions of this project. So sitting on top of all of them is this question, broad question: what is the impact of Pacific salmon community change for ecosystems? And when we say impact, we really have a subsidies perspective here. What does this mean for subsidies? And the first sub-question then is, what are the magnitudes of continental scale marine to freshwater subsidy fluxes? The nutrients we're considering here are nitrogen and phosphorus, as you would expect. We also have the fatty acids that are commonly associated with salmon consumption, EPA and DHA. And then our contaminants of choice are the persistent bioaccumulative and toxic contaminants, the mercury, PCBs, PBDEs, and DDTs. And this is sort of a chicken or egg thing. They're measured. These are the contaminants that are most commonly measured in salmon tissues because of their persistent bioaccumulative toxic properties, but these are also the ones that we could find data for. So it worked out. The second question, and I'll just say that the first question is sort of our first principles question. Like what are the numbers? No one's done this before on this scale, so we want to do it. But then we can use those numbers to ask some other questions. And the second question is, how have shifts in the Pacific salmon community altered subsidy fluxes over both time and space? When we talk about shifts in the community, we're looking at not only escapement runs, but also sizes of fish and the relative abundance of the different species in the community. We're again considering this 40-year time period. And then we also broke the continental scale analysis into four sub-regions: the Bering Sea, Central Alaska. I have a map that I'll show when we get to this part, Southeast Alaska. And then we paired British Columbia and the West Coast of the United States together. And then finally, our individual species disproportionately delivering a brighter side or a darker side subsidy profile. People working at this interface of sort of ecotoxicology and ecology have started calling contaminants dark subsidies. David Walters sort of coined this term in, I think, a 2008 publication. But we drop the assumption that they're beneficial to the recipient system, but they're materials that are being moved from one, they're being subsidized into a system by this process. And the reason we're interested in this sort of disproportionate question is because we know that the 5 species of Pacific salmon occupy distinct trophic positions. And that's important when you think about contaminants that biomagnify, because trophic position can be a key predictor on how high concentrations get, depending on how far up they move in a food web. So let's just start with question number 1. This is our first principles question. What are the magnitudes of continental scale marine to freshwater subsidy fluxes? And the answer is a lot. Pacific salmon are moving metric tons of nutrients and kilograms of contaminants from the ocean to freshwater systems each year. Conceptually, we calculate this simply as the whole body contaminant concentration of a fish times the weight of that fish, which gives us the load, the subsidy load per fish. And we multiply that by the number of fish, and that gives us our sort of biomass-derived contaminant flux estimate. It's not as simple as this. There's a sophisticated ecological model sitting under all of these results. You can just see then that the scales are vastly different. Nutrients are coming in at a much higher rate than contaminants are. And we can all be rest assured that salmon, as they move, are bringing way more good things than bad things with them. What is this? How do we conceptualize this scale? Well, the amount of nitrogen, for example, that all the salmon are bringing in on a yearly basis is the equivalent of 67 blue whales and 0.0005 blue whales in terms of contaminants. That's hard to wrap your head around, but David Walters, if you know him, has a beloved bird dog, and he's like, oh, wait, that's about the mass of my bird dog. So there's one Mabel for scale for PCBs as your counterpoint for blue whale transfer. And if you added these together and summed all of the subsidies and then said what proportion of all of these subsidies are transported by the various species, we can see that pink salmon are really taking the lion's share. They're transporting more than 1/3 of all of these subsidies. Okay, so then the second question is, how have shifts in the Pacific salmon community altered subsidy fluxes over time? We know that the community is changing. We have this data that shows this really nicely. We're using both weight data and abundance data to give us changes in escapement. And escapement here, the measures of escapement are in biomass, and the different species are indicated by different color. You'll see a lot of plots in the coming slides that have this structure. So time is on our x-axis, and concentration or magnitude is on your y-axis. Oh, these are really blurry. I'm sorry about that. So as a result of this, we know that over these last 40 years, we've seen a huge increase in the amount of subsidies. And we break this out by subsidy type, but the trends are very similar for all of the nutrients and all of the contaminants. We're seeing an increase in the range of 17% up to, I think, 31% is the highest percent increase we're seeing. Abundance of pink salmon in particular is really driving this trend. There are so many more pink salmon that they are counteracting all of the decreases in other fish, the other species populations, and decreases in their size. And if we layer this with a space question, here we're just looking at the map now, here are the 4 regions. And to just a quick way of thinking about the differences between the 4 regions is that in the Bering Sea, sockeye salmon are really a substantial portion of the population. And the other 3 regions that we consider, pink salmon are dominant. And so if we look at just total escapement, here we're looking at the biomass of Pacific salmon escapement, we can see that in the systems that do not have a huge proportion of pink salmon in their community composition, we see a decrease overall. So we see in the Bering Sea alone, we have a decrease in biomass transfer. In all of the other systems, we have a pretty remarkable increase over this 40-year period of time. And you can break that out by species, and it's really, if you could read these, it would be illustrated pretty clearly that this decrease is driven by the decrease in sockeye salmon in this system, whereas everywhere else, the pink salmon trend is the increasing trend that's driving that change over time. And this is paired really nicely with the nutrients data, which has similar shape. And here the nutrients data are broken out. Each of the colored lines is a different region with the total or the sort of continental scale pattern in the top black line. And so you can see that if you break out nutrients and contaminants in every region except for the Bering Sea, we're seeing a pretty remarkable increase in subsidy flux. Bering Sea, though, is losing both nutrients and contaminants as a result primarily of reduced sockeye salmon abundance and the fact that a lot of pink salmon don't end up in this region to counteract that loss. And then our final question is, how do the 5 Pacific salmon species compare as better or worse subsidy delivery systems? And this is a 2-layered question. The first is kind of at the organismal level, what is the potential for each of these species to transport beneficial subsidies being nutrients and non-beneficial subsidies being contaminants. And you can see work by others in the past has demonstrated pretty quickly or pretty nicely that there is this gradient in trophic level of the species paired with our biplot of stable isotopes. And this maps on, it's supposed to be colored, to the nutrient and contaminant profiles. If you could see the colors here, what you would notice is that Chinook have the highest concentrations of all the contaminants that we're interested in. So they're bigger body size, they sit at a higher trophic position. Chinook salmon have more subsidy flux potential than pink salmon. They sit at the opposite end of the spectrum. They're the lowest trophic position. So that gets you thinking, as these communities are changing, Chinook and sockeye and coho are decreasing in number, pinks are increasing in number. What does that mean for relative contaminant and proportional contaminant transfer? And this is our answer. So here you're looking at proportion change over time on the x-axis, and here's the different proportions up, from 0 to 1, broken out by species for all of the contaminants. And what I want you to notice first is the pink salmon trend, which is shown in dark purple at the bottom, have increased in the proportional transfer of all subsidies. Contrast that with Chinook, where their proportional influence over nutrients was never very high, but over contaminants has decreased really substantially over time. So then let's layer that. If we add up all of the data in here, add up all of the contaminants transferred each year and all of the subsidies transferred each year, and then break that out by what proportion of all the contaminants does each of the species transfer, and what proportion of the nutrients does each species transfer, you get this this hill figure, as I call it. And I put a backdrop on it so you could see the time series. But what we see is that Chinook and coho are proportionately more influential to contaminant transfer than pink salmon, which show a very clear signal as being primarily nutrient transporters. This is pretty interesting. The other thing that's interesting to see is that over time, we move from the 1976 in yellow to the dark blue color, which is our most recent time point. we see differences in these two species. Chinook are moving towards a more sort of nutrient favorable profile, and coho are demonstrating the opposite trend. We don't understand what's going on with coho so much, but for Chinook, we attribute this to the pretty remarkable decrease in Chinook body size. Average Chinook body size has decreased by about 25%. And here's just a summary of everything I just said, but the answers to our questions: tons of materials coming in, much more nutrients than contaminants. Pink salmon are really influential to this process, the most influential species of the 5 we considered. As a result of changes in the community composition of Pacific salmon, escapement biomass is increased 37% higher in 2015 than it was in 2017, and that translates to a really big increase in subsidy biotransport. That's also increased by about 30%. And regionally, those increasing trends are consistent everywhere except for the Bering Sea system, where pink salmon have not exerted as much influence as they have in the other subregions. And then this final question, which I think is maybe the most interesting one to emerge from this work, is that pink salmon, which occupy a low trophic position and can't biomagnify contaminants to the same level, are disproportionately delivering nutrients. They're also the species that's taking over community composition. And as they continue to dominate the Pacific salmon community, we expect that these nutrient contaminant ratios will continue to increase. So I think overall, this is a good news story. Subsidies are not on the decrease. This ocean to freshwater transfer of subsidies is robust. And as a result of Pacific salmon community changes, we're seeing fewer contaminants than we did in the past. These questions about community composition and contaminant movement in food webs are motivating a lot of questions in my group. So I just want to highlight a couple upcoming projects we're doing. We're just kickstarting now a PFAS-based project asking similar questions, but at a much smaller scale. So in a stream food web, how does PFAS concentration and composition of the mixture change with trophic transfer, and then also flux out of an aquatic system into the riparian system? And we're asking similar questions about sort of how the phytoplankton community changes and what that means for trophic transfer into shellfish in the Long Island Sound coastal ecosystem. And so just to wrap this up, I hope that the projects I showed demonstrate why I think that this thinking is valuable, and what I value most about it is I think that it's a really nice framework for thinking about global change. If we focus on changes in communities as drivers of contaminant transfer, we get to ask much more interesting questions. So with that, I'll wrap up. I hope I'm okay on time, and thank you.
Great, thanks, Jess. I want to remind folks that are in the virtual audience to put your questions in the Q&A, and I will draw from those questions, but we'll first, since there aren't any yet in the Q&A, if anyone here has any questions. Yeah, Peter, go ahead. And Jess, if you can remember to repeat the question.
I'm going to try, yeah.
So that's a wonderful talk. And
thank you.
This is the light, the dark side of contempt. It's just great. Bird dogs, you know, not all that familiar with. So is it a lot of contaminants or not? Like, so is the ocean transport a significant part of the load of contaminants that the salmon get? And because the other thing, not knowing anything about salmon, is that I noticed that they all come out of hatcheries.
Yeah.
And so, like, how much do they come in there, and how much they get from the ocean, and how much do they get from smelting plants or whatever, or other sources? Right.
Okay. I'll start with part one of the question, which was, let me paraphrase. How do we think about this amount of contaminants? Is it a lot relative to what we know about contaminants? Is that a fair assessment? Okay. Really good question, and I'm glad you brought that up. When we think about contaminants, is the amount of contaminants really the best benchmark? A little bit of mercury can do a whole lot of damage. So relative to the damage potential, that's a really good question, probably something we need to pursue going forward. I don't have a good answer for you. But the other thing that I like to point out in the paper that we're writing now is these contaminants are also really persistent in these systems. So if we're just looking at annual flux, that's not really painting the picture. If they're coming in annually, but a lot of high percentage of that mercury is staying in, say, a stream bed or being transferred to the riparian food web, what is the cumulative loading over time? And so we actually calculate that out for the whole 40 years. Over this 40-year period, this much mercury has come in and this much PCBs has come in. So yes, I actually think that the contaminant concentrations are substantial, even though they're totally outweighed by the nutrient concentrations. We need to be thinking about sort of the so what part of that, and is that amount of mercury really impactful, or the amount of PCBs impactful?
So are you going to do some mass balances for the salmon population and for some of these watersheds?
That's the next phase of the project. Yeah. The second part of your question, help me remember.
Well, that was mostly it. But
Okay.
in terms of the salmon, like, so a salmon picks up a load of mercury.
Yes.
Like, how much of this is getting from the ocean? How much is getting from the ocean? But that's a.
Yeah, we think all of it, more than, yeah. So like, I think it's been estimated that 99% of all the mercury in a salmon is coming from the ocean. And in this analysis is all natural spawners, so we don't have hatchery fish in this analysis. So the hatcheries are another factor that we excluded because there are enough differences there were enough differences to warrant that distinction. Yeah, good questions, thanks.
Great. I have a question from online.
Okay.
Vicky Kelly asked, what is it about the green algae that allows them to absorb selenium so well? And are all green algae the same?
Yeah, should I repeat that question?
Yeah, no, you don't have to repeat it because
Okay.
I have a mic. Yeah.
Good. So why are green algae so good at this, or so bad at this selenium uptake problem? I made a quick reference to the fact that organisms vary in the amount of selenium they require, and that's tied to the size of their selenoproteome. So the number and abundance of different proteins that require selenium function. Primary producers, and green algae in particular, are estimated to have the highest number of all organisms that have been measured to date. So we think it is just that they need a lot, and so they're able to take up a lot. We don't understand completely selenium transport. Some studies suggest that process, that uptake into algae, is saturable, and others say it's not. So we don't know exactly that. Some studies say actually a lot of the selenium that's coming in through the base of the food web is adsorbed, not absorbed. But does that matter for something that is eating that algae? They're not brushing off the surface, right? They're eating the whole thing, but the chemical speciation might be different. So there's a lot of complexity in that question. It's a really good question. It's super interesting for follow-up.
Great, thanks. I saw Jane's hand go up first, so Jane.
Thanks, this is super interesting. So I'm two questions-ish, but I'm surprised that like with the migration back to these hatcheries, how come there isn't this just like continuous accumulation of contamination if they're dying there and over and over again, or is it just that the water moves it on down? And then also if you could just touch on like why is pink salmon doing so well?
Yeah, good question. Okay, so the second question
So
I'll start
repeat
with,
those. Yeah.
is why are pink salmon doing so well? And there's some hypotheses, and I wish I could answer that question, but I'll refer you to our colleague Greg, who's written a ton about this. I think the leading hypothesis is that they have just sort of a competitive advantage in the systems, and they're out competing for resources in the ocean, but I'm not sure that there's actually a firm answer yet. The second question, or the first question that I'll take second, is why is there not just this huge accumulation of contaminants in these recipient systems, right? So just to remind the audience that the fish that we are interested in are the ones that go back to the streams, so the actual natural systems where they were born, where they hatched. I think the answer to your question is there is quite a bit of accumulation in those systems. It does get distributed a little bit. So like bears eat a good portion of the amount of contaminant, the amount of salmon that are coming into these streams. They get recycled. Some of them probably get sequestered in the sediment of these systems. And so maybe they're only going to be released after sort of a disturbance-based event. But some of them are probably kind of lost to the system, but they do move around a little bit. But there's also concentration in this sort of contaminant transfer work. There's some really nice case studies about seabirds and how they bring some of the same contaminants into these, like their roosting sites, and they drop them from their guano in these systems. And there's just like a huge increase in PCB concentration as a result of that process. So they concentrate them. And there is, I bet if you went to these systems and took like a sediment profile, you could probably see a history of that. I don't think that work's been done. Some of the earliest work actually did do that. And it was a PCB paper. Can't remember the author. I think it was in Nature or Science that showed that the sediment profile of PCBs matched the PCB profile of incoming salmon. That's how they first, I think that was the first paper to show that salmon are bringing back contaminants too.
Great, thanks. Other questions? Chris, I saw you had your hand up.
Yeah, I had a follow-up on the the algae was higher in April or something like that.
Yeah.
Do you have a hypothesis about what's going on there, or why is it?
It's, well, okay, so the April coincides with snowmelt. So it could be that these algae are just primed seasonally to be growing. So there's this like robust growth spurt, and that is related. It could be a community composition thing. We haven't measured sort of the various species that are in that community, but if, and I have a hypothesis about this that we're trying to get funded, is if the community is changing over time and that community change is associated with different proportional representation of species that require lots of selenium versus low selenium, does selenium uptake potential follow that trend? Right, so if you have in April, a lot of the phytoplankton or the green algae community is represented by species that need a lot of selenium, is that why we're seeing so much uptake? But we don't know. It's something that we need to be teasing apart.
Evan, go ahead.
Yeah, really interesting talk.
Thanks.
It's really interesting work, particularly the salmon stuff is fascinating. I was wondering if you could comment if there's been work comparing some of these salmon spawning grounds in terms of their contaminants or even just the food webs of the tree dynamics to comparable streams that lack salmon spawning grounds for whatever reason. So like try to parse sort of like the end effects of what this.
Yeah.
Don't forget to repeat the question.
Yeah, I
thanks.
know I forgot to do it on the last one, sorry.
Yeah.
The question is, has there been work to compare streams where salmon do return and don't return to look at their contaminant profiles and maybe even the sort of the effects on the recipient system? Other than that one study that I mentioned as sort of like one of the first studies to ever look at contaminants in salmon and how they match the sediment profiles of salmon recipient systems, they did have a reference system to compare to that was not a salmon receiving system. That's the only study I can think of off the top of my head that's done that work. But we hope that this study, because we included nutrients strategically so that ecologists will pay attention to it, that it'll motivate some of that kind of work. And I would love to be involved with that kind of work if there was an opportunity.
Great, thanks. Other questions? Yeah, Winslow.
I was curious generally, but specifically came to mind with the example of the green algae. Are there temporal lags in concentrations across trophic levels? So if your stream water has a concentration of X, is the stream water a month ago a better predictor of green algae?
Yeah,
concentration?
very good question. The question is, are there, maybe if I can rephrase it, what are the different time lags and sort of exposure and uptake and retention, right, at different levels of the food web? Is that fair? And specifically about the green algae, would the water concentration from an earlier period of time, say a week ago or a month ago, be a better predictor of the concentrations we're measuring? We didn't answer that question in our sampling, but we had the water concentration data and we did ask that question. If the concentration of selenium is high a month in advance, on what time scale are algae responding to that water concentration? And we think it is pretty right on top of each other temporally. Yeah, but another question that is really, I think, important. So if we do continue to monitor just the water concentration, because there's a lot of energy and infrastructure around doing that already, and it's sort of in our systems, we wanted to keep doing that. What are the ways that we could make it the most useful predictor of what's happening in the food web? And maybe that is one of the questions we need to ask. If we see this, we can predict that the fish are going to, in 3.5 months' time, have the peak concentration that we just observed in the water column. It's a really good question.
Yeah, go ahead, Jane.
So you mentioned your chemistry pair with the contaminant. I think it's like a very, very cool data set. Is it just kind of a linear, like more nutrients, more contaminants, or do you see any interesting comparisons, and do any of the nutrients predict, like, could you just look at nitrogen content and say we're going to have this much of this?
Super good question. Didn't even think about that. So the question is a couple of parts, but I'm going to focus on the last part, if that's okay. Are the nutrient and contaminant signals or patterns linked so that if you wanted to just measure nitrogen concentration, you could get a pretty fair estimate of the other constituents? I don't know, but that's the first thing I'm going to do when I get back home is look at those ratios. Do they match over time by species? Because that would save a lot of money from an analysis perspective too, right? That's a good question. In terms of the temporal trends and what we saw in these systems, we don't have enough data to ask the question about how contaminant versus nutrient patterns are changing. We do see some indication that they're decoupled. So as fish are getting smaller, for example, they're bringing probably the same amount of proportionally nutrients, but they're disproportionately less contaminants because of the trophodynamic behavior of contaminants. But we don't have the data positioned quite right to be able to ask that question with this data set.
I'm going to ask a
Jump
question,
in.
if that's okay. So I note that a number of the contaminants that you looked at for the salmon, PCBs, DDT, so you were looking from the 1970s to present.
Yeah.
Many of those were banned in the 1970s, PCBs and DDT.
Yeah.
And so we see this so like it's an important thing, legacy effect.
That's right.
And do you have any thoughts about how long this legacy will continue to persist?
Ooh, really good question. So yeah, I forgot to say too that when we had to go from sort of 1925, we had to cut out the 1925 to 1975 time period from our analysis. But that ended up being okay because one of the contaminants we wanted to consider were the PBDEs, and they were just coming online in that period. So it actually worked out from a data matching perspective. And so you're asking what is, a lot of them were banned in that period. A lot of the contaminants data we have in this paper is actually from the late 1990s or into the early 2000s. So the concentrations are reflecting a little bit of that lag. There's been some really interesting work. So yeah, to think about legacies, there's been some really interesting work to show that even for mercury, for example, emissions have been substantially reduced. We've gotten mercury emissions under control, but concentrations in food webs aren't necessarily decreasing. And so there are some of these extrinsic mediators now that are kind of taking over, and some of that is related to food web composition. So I could hazard a guess that these are going to be, their legacies are going to be pretty long, and we're going to be seeing them, and we might even see higher concentrations because now the food web's taking over.
Yeah, okay, great, thanks. Well, we're just about to the hour, at the top of the hour. So thank you again so much, Jess, for this really interesting seminar. And thanks everyone for attending online and for attending in person. And we'll see you next week. So thank you.
Thanks for having me.